Welcome and thank you for standing by. At this time, all participants are in a listen-only mode until the question and answer session of today's conference. At that time, you may press star 1 on your phone to ask a question. I would like to inform all parties that today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the conference over to Irene Ihir. Thank you. You may begin.
Thank you. Hello, I am Irene Ihir of CDRH's Office of Communication and Education. Welcome to the FDA's 40th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to the development and validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Timothy.
Thank you, Irene. And hello, everyone, and welcome back to our town hall today. I'm going to start off on a somber note, a remembrance for a pioneering COVID test developer who we recently lost. So, Dr. Andrew Brooks was a research professor at Rutgers New Brunswick School of Arts and Science in the Department of Genetics. He also led the creation of the first EUA-authorized saliva-based test and the first home collection of saliva. And reportedly, reported in the news now, at least on the Rutgers site, more than 4 million of those tests were performed between authorization and now. So it's really sad to see somebody who was able to advance testing in this way to be lost to us. He passed unexpectedly on Saturday, and leaves his wife Jill and two daughters, Lauren, Hannon, and Danielle. Sorry, it's been a long fight. Anyway, we're very thankful to him for his work, and we'll remember him always. Okay, pull it together, Stenzel. I wanted to give some heads up on mutations. This continues to be a very hot topic, and I think it's ideal for those that already have an EUA authorized test, and those that are developing tests to consider the variants. We moved to authorizing single target assays relatively early in the pandemic, with the knowledge at that time that single target assays, we had not seen mutations that would impact them. We are starting to see mutations to impact tests, and obviously single target versus multiple target, targeting multiple regions of SARS virus. And there may be a performance difference going forward. So we'll certainly stay vigilant at the FDA's side on monitoring this. But we do want to put a shout out to developers to certainly be considering this, and they and to work with us as looking ahead at how we can maintain the highest performance possible for COVID tests. So molecular tests, that includes probes and primers, We're obviously open to talking to developers who want to make updates or that are in development about how best to do this. So no change in recommendations at this time. Just a heads up. We think that antigen developers and serology developers should keep these variants in mind as well. Numerous of the serology tests do target the spike protein, which can alter, and at least one of the antigen tests does target the spike protein. But of course, there can be mutations that could affect other genes, other targets within the virus. So with that, we'll go into questions. Thanks so much.
Operator will now take questions.
Thank you. We will now begin the question and answer session. If you would like to ask a question, please press star 1, unmute your phone, and record your name clearly. Your name is required to introduce your question. If you need to withdraw your question, press star 2. Our first question comes from Hannah Gabrielli. Your line is now open.
Hi, thank you for taking my question. So I came up and asked questions a few weeks ago about breath tests that we're developing, and I wanted to thank you for your answer then, because I actually received the recommendations for breath test developers and resubmitted a pre-EUA according to those considerations. I wanted to ask today, because this is something that I haven't found anywhere, what is the required sensitivity that you are asking for breath screening for COVID and respiratory viral infections?
Sorry, I didn't catch that last part. What is the what?
What is the sensitivity that you are requiring for breath test screening?
Yeah. So this is an important question, and in fact, our team, I've charged our team with coming up with targets for this. They promised them to me today, but probably by the end of the day. And
so we haven't received any feedback yet.
So, you could imagine, and I'll just talk a little bit philosophy on a non-standard sort of test. First of all, we're very open to novel tests. We have no idea if breath tests, truly can perform, but we're totally open to it. We do want to have them validated appropriately, and since they're a newer technology, we're likely to ask for more than sort of the standard technology samples. So I don't know if they gave you sample numbers yet, but we're working on that as well. You could imagine that the breath test might be used for either diagnostic purposes and or asymptomatic screening, surveillance. The FDA has stated that for this pandemic, we don't intend to regulate for surveillance. So the other thing about breath tests, because there may be other breath test developers out there, is that we are concerned with potential carryover from one patient to the next. So that will be an important consideration of the safety, and that may not be in the recommendations yet. But I think there's going to be challenges with whether a developer wants to pursue symptomatic claims or asymptomatic claims. Some there's some thought, I'll just be open, that, we don't know if symptomatic or asymptomatic claims are necessarily the best claims to go to first, because if someone has symptoms, ideally they might need a test that that has a longer proven track record. On the other hand, we have a huge need to expand testing into the asymptomatic screening population, and so that might be the more ideal population. There's no, we're open to all comers here. So if you want to come in and you want to pursue symptomatic claims, we'll look at what that looks like. But I do see the sweet spot potentially for this technology is if it can perform in the asymptomatic population, that it could really address some of our needs to really increase not only the amount of testing, but the frequency that everybody can get tested with. So we'll want to look at in whatever population, we'll want to look at what is the sensitivity or PPA relative to a molecular test, high sensitivity molecular test, and also the specificity or NPA. So does it call any false positives? Now, it could be that for benefit risk calculations, we may decide that positives should be double checked with molecular tests. So that's some of the thinking that we're looking at right now. However, if the NPA if the PPA and NPV negative predictive value is extremely high for, say, people in the asymptomatic population, that could really be a win. And I'm just thinking through all the calculations right now. So I think we're going to ask for more positives and more negatives than we typically had, and we're working with our statistician on determining what our recommendations are going to be there. So stay tuned. I think we'll be able to update our thinking hopefully by next week, so that we can share with developers our latest thinking on that.
Thank you so much.
Mm-hmm.
Our next question comes from Josh. Your line is now open.
Hi, thank you for taking my call. My question is, what would be the process if, a developer of a EUA authorized molecular test wanted to make a change to the primer and probes, perhaps in response to a mutation, or proactively to make the test more resilient to mutations by adding a second target? Would that have to be a whole new application that would have to go through the same queue, or can a change be made to the existing one, or what would be the process there?
It would be potentially a supplement. It kind of depends on what you're going to do with the original test. If you're going to take it off the market when you have a new authorization, then there may not need to be a separate authorization. We're going to want to look at how the changes affect your accuracy. So, and you could imagine different ways to account for mutations. You could use degenerate primers and probes, and we would just want to make sure that, however you design those, doesn't decrease the sensitivity of the overall reaction. There are different ways to go about that. And then, if you are adding a new target, we do want to make sure that target is going to be accurate. We know the level of that accuracy, and so that would be, pretty much all the key elements of development, which would be, if it's just for symptomatic claims, then 30 positives, 30 negatives in LOD, and in the cross-reactivity and inclusivity testing that we asked for.
Okay, and so it sounds like if we were to take the existing test off the market and replace it, that this could potentially be a faster review process rather than submitting a whole new application.
If somebody is trying to improve the safety due to the variants, we're gonna make that a priority review. So I think that's something that we haven't set as a formal priority, but very clearly that's an important thing to do.
Okay, thank you.
Our next question comes from Citrali Kulani. Your line is now open.
Yeah, hi. We're developing an antigen test, lateral flow, and I just had a question about combining a prospective study and a retrospective study. So if we follow what's on the EUA, like provide retrospective samples, and then along with 5 positive prospectively collected, is that considered okay, especially for a point of care claim?
That's our current thinking, yes. So
okay.
Then there would be a post-market commitment.
Yeah.
If everything looks good and we can authorize, there would be a post-market commitment to figure out.
And then do we have to provide any fresh negative samples?
Yes. So if you're going to do a prospective collection of 5 fresh samples, you're also going to collect negatives in that. So, that shouldn't be any problem for you to collect, the 30 negatives or whatever the minimum negatives is pre-market.
Okay, so for negatives, mostly you'd want them to be covered pre-market, and then post-market it'd be mostly on the positives.
We don't think that's going to be too burdensome because the negatives are going to be relatively prevalent. It's going to be the positives that you're going to have to wait on. So, I would just, set up your clinical study and collect all the negatives or that you need or that you're doing in your study along with looking for the first 5 positives.
Okay, and then on our EUA application, we would just state that we have an ongoing prospective study.
Yes. You can let the team know that you're working on getting those prospective fresh samples.
Okay, thank you.
You're welcome.
Our next question comes from Kirsten Banker. Your line is now open.
Thank you for taking my question. I was just wondering what is the role of tunnel government service staff in the review of EUA applications and how this affects the FDA review process and the target review time?
I'm sorry, could you ask that question again?
Oh, sure. So I was just wondering about the role of the tunnel government service staff in the review of EUA applications and how this affects the review process and review timelines.
I think you may be referring to some contractors who we've hired to help with reviews. So the latest surge doubled. Prior to the surge, we had brought on contractors, and then more recently, we had, with internal resources within the center at the FDA, we doubled the resources on COVID. And so all of those additions have allowed us to make a lot more decisions more quickly. Now, some of those decisions aren't always positive decisions, and we certainly don't advertise and post negative decisions unless we're removing something from the market.
Okay, thank you.
Mm-hmm.
Our next question comes from Kay Taylor.
Yeah, thank you and your team for the opportunity these town halls provide the attendees. My question is regarding clinical trial design for at-home antigen tests. In particular, my question is the collection of the nasal swab samples for both the test device and the reference PCR device. The test sample device, nasal swab, would be self-collected by the representative at-home user, and the reference device sample would be collected by a healthcare professional. Does FDA have guidance on the order of the collection, meaning should the order of the nasal swab collections be randomized, or would FDA expect the self-collection to occur for the test device first and then always followed by the healthcare professional collecting the swab for the reference PCR test?
Yeah. So again, are they both anterior nasal swabs?
Yes.
Okay. So, there's sort of ideal, ivory tower sort of answer, and then I'm going to give you the more practical and the answer that I think is best to follow, and the fairest route for the candidate device. This is fairly simple, although hopefully I don't mangle the direction here. But yes, you do want to randomize, okay? But you also want to make sure that the candidate device isn't at a disadvantage here. And a number of studies have been performed this way, and we don't think standard of care is being significantly harmed if the comparator test is, as we request it, is a high sensitivity molecular assay with a separate extraction, with an extraction step. And we urge people to look at our FDA reference panel data to identify the assays that are high sensitivity. Then what you do is this can be, this is self-collected though, isn't it? Oh gosh. All right, this this is a challenge. I haven't gotten this question before. How do you randomize a self-collected swab? How do you not bias the self-collector with this? Does it have to be a healthcare professional who's doing both swabs, or one swab, or can it be the patient candidate that is collecting both swabs?
We presumed with the reference PCR test that we would have to follow the instructions for use, which typically call for a healthcare professional to collect.
All right, so you're not using an at-home kit. You're not using an at-home kit for this.
Correct.
You're bringing them. Okay.
So, but you're asking about whether this would be for home collection that you'd be trying to validate, right?
Yes. So the test device would be an at-home antigen test, but the reference comparator would be a professional use,
right. Okay.
A professional use PCR test.
Right. I think we typically ask for the layperson collection to go first, so that the trained user isn't inadvertently training the layperson on specimen collection.
Okay.
The challenge with that is if the patient collects the swab first in both anterior nares, which is typically the procedure, right? You could leave significantly less virus in the anterior nares for the candidate, for the comparator test.
Right, it would introduce bias there.
Yeah. So I've been typically thinking of having this be a dual self-collection procedure, but when your comparator is one that doesn't have a self-collection option, that presents a challenge. I think it's best that we
be a delay. Sorry.
Yeah, you could collect after a period of time. So you could sample. The patient can sample their anterior nares first and wait a period of time. And we have discussed this months ago with the RADx program, and about how they might do this. And we talked about some time periods, but we didn't know what the best time period was. One would be 15 minutes, I think. We discussed up to an hour. So that would be actually probably the best way to do this, is to, thank you, Toby, for reminding me of that previous discussion on this topic. So that is one clear option. If that option doesn't work for you, I think we're going to have to take this offline and think about this more. So I would approach either your current reviewer or send a question into the template email box. But that would be the best way to do this. We do allow, if a single individual wants to collect both random swabs at the same time, is to swab. One swab swabs one nare, the other nare, and the other swab swabs the other nare, and then you swap and do a second collection in both nostrils, and then you randomize which swab goes to which test. But in this case, and Toby's right, we're trying to not train the user, and if you were randomized 50% of the time, you potentially would be informing the user. Or if you're trying to do a balanced swab, you'd be training them during every procedure. So those are the options. If you want, I think the first option is have a washout or a restoration period. They do the self-test and have them wait around for a period of time, probably no less than 15 minutes, probably an hour, but we have no data to support that time period, just that we want you to delay. So you probably ought to double check that delay with our review staff, okay?
Okay, thank you, Dr. Stenzel.
Thanks, Tony.
Our next question comes from Catherine Miranda.
Hi, thank you for taking my question. My question is about sample collection, and we would like to know if an EUA-approved molecular diagnostic test is authorized for nasal swabs. Do the nasal swabs have to be collected by a healthcare provider, or can they be self-collected in a healthcare setting under the direct supervision of a healthcare provider?
So it depends on the authorization of the EUA test. When we use it as a comparator, it's important to use it as instructed in the IFU and not to go off-label. If you have to go off-label, we would need to look at, whether that was an appropriate modification, and may need some validation data depending on the modification. But it does present us, it would delay our reviews if you're not following a comparator assay as instructed, at the very least. And I can't promise that we would allow it.
Okay, thank you.
Our next question comes from Elaine Allen.
Hello. Thank you again for taking my call. My question is in relation to, in our EUA application, we have met the minimum EUA requirements, which include samples from U.S. donors only. We are expanding our clinical agreement study now to include outside the U.S. samples. Would you accept that data that includes OUS samples, whether that is for the post-EUA data update, or if we submit an amendment to our data in the IFU?
Remind me again about your device. What is it specifically, and is it a point-of-care device?
It is not a point-of-care device. It is ELISA, serology ELISA.
Okay. Well, it's probably amenable for testing at the NCI. So we do an initial review.
Amenable for what? I'm sorry?
Oh, so it's ELISA for serology or for antigen? What is it?
Serology.
Serology, yeah. So the ELISA test that can be tested at the NCI, at the Intergovernment Interagency Testing Program, you would have your own testing that you do, but it would also be double checked if it's amenable at NCI. So we'd have that data as well to support the application. So we have not required for such a test that the samples be U.S. based. We like to see U.S. based. For point of care, I'll just say, because I brought it up, it is important that the setting for point of care testing, mimic the U.S. And it's ideal if the point of care studies can be in the U.S. because how point of care testing is done in the U.S. It can be substantially different from other countries, and we certainly see point of care studies done in central laboratories with trained workers. So that's obviously, even though it might be a device that's amenable to point of care, that's not what we're looking at. We're looking at can an untrained healthcare worker who's not a laboratorian perform the test and get accurate results. But anyways, back to your question. Yeah, we have no requirement for U.S. samples and U.S. Testing, although it's ideal.
Would there be any benefit to us tracking the regions of those samples?
That sort of thing may only come up if we see funny results and we're trying to figure out, what's going on. So it's best to capture as much relevant information as you can on these samples because we often want to know where they were collected, when were they collected, and how were they stored if they're banked. All those questions are really important. And then all the relevant, important information is important as well. But, I anticipate with the variants progressing, our evaluation of all the tests is going to be more challenged. And the validation of the test might be challenged as well by variants. So, so our experiences early in the pandemic may be different for serology and other tests than they are currently or in the future. So it's just unfortunately something that we're all going to really need to stay entirely on top of.
Yes, we're keeping our ear to the ground on that one as well. So thank you very much. That was very helpful. I appreciate it.
You're welcome.
Our next question comes from Misha Lee.
Yep. Hi. Thank you for taking my question. Following up on the topic of mutation testing, I know it's indicated in the last town hall that FDA is proactively reaching out to already authorized molecular developers to conduct additional testing. My question is for manufacturers that will be submitting their new point of care nuclear protein-based antigen EUA, is there any additional variant testing required as part of the initial EUA submission?
So let me, ask the question of you, because I am going to reach out to all the EUA authorized antigen test developers. Can you use, say, heat inactivated antigen or virus and or irradiated virus from BEI? Because if antigen tests those antigen tests that can use this, and of course, a lot of this is going to be comparing to the original Wuhan strain that was deposited at BEI. We typically think of that as everything comparing to that. But if an antigen test is amenable for use of inactivated virus, that makes all of our lives a lot easier as far as testing reactivity and inclusivity. And it's my understanding that the UK variant has been deposited at BEI, and I'm just going to be double checking with them that they are planning to inactivate that. So We haven't figured that all out yet. We just know that at least for key variants, we're gonna start wanting to know, if there's any loss of sensitivity with those variants, and we're trying to figure out the very best way to do this. And using live virus in a lab is not ideal. But if inactivated virus can be used, then potentially panels can be distributed to developers and to tests that are already on the market. So stay tuned is the bottom line. But obviously the mutations, variants of interest right now of key public health importance are the UK variant, the South African variant, and now what's called the Brazil variant.
Okay. So in terms of testing the type of variants, are those the only two that we should be testing, or what is the best way to keep current on the type of variants?
So we are working through this process. So we, in all likelihood, will be updating template. In all likelihood, we'll be updating letters of authorization of these going forward. And how best to do this. So, and we're just at the beginning stages of thinking through this. It would be ideal if we can be using in silico analysis to some degree. We use it, of course, for molecular, but if we could start using it for antigen and serology tests. We are going to start beginning to ask developers how they think they can monitor variants of concern. And obviously I mentioned 3 variants, South Africa, UK, and Brazilian. But there could be, and those are important because they appear to be more infectious and could have potential impact on vaccine efficacy. So, but there could be one or more variants out there that in accumulation could start to depress the sensitivity of assays. There's even some cases we're thinking in serology examples, particularly for neutralizing antibodies, where there could potentially be false positives, right? So you have neutralizing antibodies to, a prior infection, but not to, some new variant. So all I can say right now is this is one of our top priorities, if not the top priority right now for our office and our center, and I believe the agency as well, to try to figure out how to best do this. So certainly we've been having lots of conversations. So in all likelihood, as I said, we're going to be asking or doing a template update, asking developers, what are their thoughts about how they can monitor for impact of variants and work with us essentially to figure this out. And whether modeling for antigen and serology tests can be used is too early for me to say how useful that is to figure out if a variant could have an impact. And then if there is a potential impact, for the molecular tests already, we have been asking select developers to do wet testing to make sure that the variants don't mess with the sensitivity of the assay. And we want to keep that kind of wet testing to a minimum if possible, because there are just so many variants, obviously.
Okay, thank you very much. Sorry, you had mentioned that that you guys are working on updating the template. So before that update, do you suggest that we reach out to the EUA email address to obtain specific guidance on variant testing?
You can, but I don't think we're ready to give recommendations right now. We're focused on we're doing it for molecular, and all molecular developers do this in the process of submitting their applications. They survey the landscape. Since we don't have a solution that's really workable yet for antigen and serology, I don't want to be making recommendations about that. But other than we want to know what your thoughts are about these. And as we develop our knowledge base and information base, and as we discover ways to do this better, we may update those recommendations. But certainly, if you want to bring it up, I'm not holding you back. I mean, I think it's a sign of a good developer that brings up these issues. I just don't want to hold back authorizations until we can figure out how to do this right. We are likely to, in addition to the template asking for what your thoughts are on how to monitor the impact of variants, we are probably going to put that into a commitment for post-market monitoring. And if you discover something, to contact us, and we'll figure out the next steps. And the other thing is, these are just thoughts. Nothing's settled, nothing's determined yet, but I think it is an important topic, and it doesn't hurt to bring this topic up as we try to work through it.
Thank you very much, Dr. Stenzel.
Mm-hmm.
Our next question comes from Stacy Hausman.
Hi, Dr. Stenzel. Thank you for taking my call. I work for a CRO, and I'm currently working with a client who is working on a rapid antigen study, and they plan to have their own proprietary transport media. They've seen some lower sensitivity in the universal transport media, so they plan to use this proprietary media. And according to the template, they plan to do retrospective samples for the method comparison, but according to the template, there is a requirement or recommendation that because of erroneous results seen in transport media, that there is the prospective enrollment of, it says 30 positives, 5 of those needed for the EUA submission, and then the other 25 can be done post-authorization as a condition. So my question is, I heard you earlier with another call saying that there were also negative samples, fresh samples required for that. And so I wanted to verify that that is correct. And then also, if they're using their own proprietary transport media, is it still necessary to demonstrate the low risk of erroneous results using transport media versus a dry swab?
So it's a new transport media device, one we haven't authorized before. We're going to want to know that it in of itself doesn't result in inaccurate results, and that using it in transport preserves the target. And you know what the temperature profile and time profile are in experiments and basic transport experiments. I'm not sure if that addressed your question. I'm not sure that I fully understood your question.
Well, if they're, okay, so in the template right now, if one uses, if retrospective samples are used for the comparison study to a RT-PCR method, the template suggests that because of erroneous results that have been seen in samples that were stored in transport media, there is a prospective study that is needed, 30 positive samples, 5 of which are needed in order for the EUA submission. And then the other remaining samples can be enrolled after. Is that correct?
That's the recommendations for point of care fresh samples versus using banked samples. So you can do a complete study with banked samples, but then fresh samples, we want to see some fresh samples because bank samples, especially frozen, can potentially alter the performance. Sometimes it improves performance of a device to do a freeze thaw. So are you talking about the comparator transport, or are you talking about your candidate device transport media?
Well, the retrospective samples will be in UTM. And so that'll be used for the comparator method. And the candidate method. But then the prospective study will be done using their proprietary medium.
Okay. Yeah. So that presents a challenge that I want you to discuss with the your reviewer or to the template's email address, because you're using a different media for validation, and the media could have an impact. So the reason to allow retrospective samples in the testing is to speed access to the market of an innovative test. And it really needs to be, everything else sort of needs to be the same. So if you have, if your test is dry swab, then we want to see if you have a collection of frozen bank dry swabs, then the fresh study needs to be on dry swab. But if it's in transport media, it needs to be in the same transport media so that we can use the data from the bank samples to presume performance of the test with only a handful of fresh samples to prove that fresh samples work pre-market.
Okay, so if they use retrospective samples in UTM, then they'll still need to do a full, fresh prospective enrollment study with their own transport media in order to submit this.
Yeah, so it's important to validate it with the transport media you're going to use for the test. And so if you're going to, so we certainly can give, a moderate or high complex authorization based on banked samples and use of UTM for transport media. But you've got a new media, and we don't know how it performs at all, and we don't know if it is equivalent to UTM or not. And so we do need a full set of data for the new media.
Okay. All right. Thank you so much.
Our next question comes from Scarlett Shaw.
Hi, thank you so much for taking my question. I have a question about asymptomatic screening as it relates to antigen tests. The template indicates that you should test in subjects both without symptoms and without other reasons to suspect infection. So as part of this without other reasons to suspect infection, we're sort of wondering what time frame would be considered appropriate to say no symptoms plus no known contact with someone who is positive. So would 21 days be an acceptable time limit, or should we consider a greater time limit for no contact with someone that's infected?
So the goal of this is not to enrich with people who might be pre-symptomatic, right? And if they were exposed recently, that would, potentially, give the candidate test an advantage and not truly test. What we're looking for is an asymptomatic population. Formally, we've designed asymptomatic as at the time of testing, they have no symptoms, and they don't have any prior positive test results or symptoms, I would think, I'm not sure if we have a specific day in mind here, but it would probably need to be in at least the 14 to 21 days. I think that's something that I can't give a formal answer on because I don't have a prepared response today. So, it'd probably be best to check with our templates email and our email, or if you already have a reviewer, with the reviewer for what the period of no symptoms should be prior to testing, or no exposure prior to testing.
Okay, thank you so much.
Our next question comes from Jackie.
Hello? Hey. Hello?
Hello.
Hi. Oh, hi, Dr. Stenzel. I have a question about getting vaccinated to include the vaccinated population in a serology or a neutralizing antibody tests. And my question in particular is on the clinical study design. I'm interested in understanding FDA's thinking on the sample size and also the acceptance criteria, because there's not that much that we know about the vaccinated population at the moment. So a lot of the information we will gather will be observational. Like it will be an observation. So trying to get FDA's thinking on how to best design this clinical study.
Can I ask a clarifying question? So I missed it probably. You're wanting to know if you can include vaccinated folks in your test, or are you looking at trying to predict if there's a good vaccine response or immune response?
Yes, we are trying. We think we can. We are trying to look for if a patient get a good vaccine response.
So that is something that we've not provided template recommendations for. It's and it's something that we would dialogue with CBER, who reviews the vaccines on. To date, they haven't been interested in sort of companion diagnostics here at the moment, but I always defer to them, and our thinking always can change quickly. Obviously there could be some benefit here, but what would you say? You would basically say after vaccination, well, you've got antibodies to SARS-CoV-2, right? Going the extra mile, that says, well, what does that mean? Is a little bit of a challenge, right? So the bottom line is we don't have current recommendations for such a study, but we are letting people know that if they want to do such a study, that they can propose a study design and we would review it. At the moment. And of course, we would not just review it in our center, the device center, but also in the biologics center.
Yes, we have a proposal, but we wanted to make sure it is sufficient sample size and access. And the challenge is that right now we don't know how to set the acceptance criteria because it is still an observational study. So we will just track patients, or not patients, but individuals who have received a vaccine over a period of different days, like at different time point every week for a certain amount of time.
Yeah, I would, again, just recommend that you put your best thoughts into a proposal and then ask the questions that are unanswered, okay? And I don't know if you've already submitted it or not. If you have submitted it, did you get responses already from us?
No, we really wanted to do a good job before we sent it over. But we plan to submit it soon as a pre-EUA format. So thank you.
Yeah, oftentimes when we have new developers coming in with new ideas, shall we say, we like to really explore those ideas with the first several developers so that we can then formulate potential recommendations that make sense. Okay, we don't. Okay. And so I think that dialogue with our staff could be very beneficial to you, and certainly would evolve in our thinking into the point where we might be able to start formulating some recommendations.
Okay. We would really appreciate the dialogue. Can we send it in an Excel format, or we probably should put it in writing, right?
Yeah. You can attach an Excel spreadsheet, but we want it's best to be in written form.
Okay. Thank you so much, Dr. Stenzel. Thank you.
And that will be our last question. I would now like to turn the conference back over to Irene